So this mini lecture is just to show you an interesting application or way we've studied glycolysis. So there's a relationship between increased levels of glycolysis in cancer cells. So remember that glycolysis is the first step in either cellular respiration or fermentation. And what we have found is that in some cancer cells, the cancer cells actually increase the rate of glycolysis. And we think most likely, I'm not sure if they know exactly where the pyruvate's going, if it's going into a fermentation process or going into the aerobic respiration. But what's important is the cells are just trying to crank out as much ATP as they can. Remember that glycolysis only nets to ATP, so it's not a very efficient process. And normally the pyruvate goes into cellular respiration to the mitochondria, where you have lots of ATP synthesis. But what they've found in cancer cells is cancer cells it says right here, will preferentially use glycolysis while decreasing oxidative phosphorylation. So they're decreasing the whole aerobic pathway and just cranking out ATP through glycolysis. This is really interesting. Glycolytic enzymes are overexpressed in 80% of all types of cancer. And this may be due to low oxygen levels inside tumors. So tumors become these masses and it's hard to get oxygen to diffuse through. So there may be a reason to increase glycolysis for ATP production. What's happening, how we study this, is that we can use radioactive molecule called fluorodeoxyglucose. So here's fluorine. And from your periodic table, you can tell that fluorine has 9 proteins and usually 10 neutrons. Fluorine 18, which is a radioisotope, which means it gives off energy as it breaks down. And we can measure this energy. So fluorine 18, this is referring to the atomic mass. So this means that this fluorine has 9 protons and 9 neutrons. So remember, changing the number of neutrons is what makes an isotope. Having something that breaks down makes it radioactive. And so what they do is they attach a radioactive fluorine, right here to a glucose molecule, and it's called fluorine deoxyglucose. So you're losing this oxygen right here and putting in the fluorine molecule. And so what happens is that here's your glycolysis pathway, and fluorine deoxyglucose, where the fluorine is right here, this big red dot, it cannot be broken down any further by phosphoglucose isomerase. So glycolysis stops right here, which means this molecule remains stable in the body. And the half-life for radioactive fluorine is 110 minutes. So what they do is they have a person drink this radioactive glucose, let it go through the body, and then they take an image of the body. And so you'll see these bright spots glowing that are radiating energy, and you can look for cancer in a non-invasive way. So just to orient you, this is the person's brain, and their brain does not have cancer, but it uses a lot of glucose. Right? So your brain using lots of energy, so the glucose will accumulate, the radioactive glucose will accumulate there. This is the person's bladder, and this is the person's kidneys. And those two organs are processing the radioactive glucose. So the kidneys are filtering it through the bladder's getting ready to release it from the body, but here are where the tumors are. So this area, this person has several areas of tumors, and we can tell they're tumors because they're high in pulling in that accumulating that radioactive glucose, because they're doing lots of glycolysis. And because they're doing so much glycolysis, but it's stopped here, the radioactive molecules accumulate in those organs, or in this case, in those tumor tissues. So, just an interesting application of glycolysis and radioisotopes and looking at them in medicine. Thanks.